Hi, and welcome to the Peter Como HD Research Symposium. My name is Erin First-Hemming, and I'm the chair of the HSG Publications Committee. I'd like to thank the committee members for their assistance in selecting the posters and platform presentations. Our HSG Publication Committee members this year are Eric Molo, Clement Loy, Sandra Skinner, and Paolo Moretti. Thank you for your excellent work. I'd also like to thank Kevin Gregory for all of his help behind the scenes. We accepted 71 abstracts this year, and I'd like to encourage everyone to view the posters in the virtual poster pavilion. As part of our annual meeting, the Peter Como HD Research Symposium provides an opportunity for researchers to share their valuable findings with the HD community. The symposium was named after the late Dr. Peter Como. Peter Como was a renowned neuropsychologist who made an indelible mark on HD-related research and care. Now, I'm honored to introduce our platform presenter, Sara Pico. Sara is a Ph.D. student at the Center for Molecular Biology in Madrid. Sara joined the Molecular Biomedicine Master program at Autonomous University in Madrid in order to start her scientific career working in neurodegenerative diseases in José Lucas Lab. There, she started investigating the role of RNA-binding proteins, specifically in HD. It was in this way that they discovered a potential therapeutic target for HD, which led Sara to develop her skills in analyzing HD patient samples, as well as preclinical studies using HD mouse models. Their work was recently published in Science Translational Medicine. Thank you so much for joining us today, Sara, and thank you to everyone that is watching her presentation. Enjoy.
Hello everyone, I'm Sara Pico, PhD student from José Lucas Lab at Center for Molecular Biology, Severo Ochoa in Madrid, and I would like to thank the Huntington Study Group for bringing me the opportunity to present this work entitled Cpv Alteration and Aberrant Transcriptopolymorylation Unveil a Treatable SLC19A3 Deficiency in Huntington's Disease. Well, first, I would like to introduce you these Cpvs proteins. And as a brief introduction, Cpvs, cytoplasmic polydenylation element binding proteins, are a family of RNA-binding proteins where Cpv2, 3, and 4 are closely related, and Cpv1 is the most distant member of the family. These proteins, Cpvs, recognize transcripts with CP sequences, which are sequences with an U-rich motif in their 3' untranslated region, UTR. Bioinformatic approaches have shown that nearly 40% of the genes are regulated by these proteins. More precisely, its function is to repress or activate the translation by inducing shortening or elongation of their poly-A tail. This cytoplasmic elongation have essential functions controlling gene expression in the adult organism, thus providing temporal and local control of translation of specific mRNAs. There is no evidence linking these proteins to cancer, diabetes, and also with autism, but the potential role of Cpvs in neurodegenerative disorders has not been fully explored. So for that, we thought that Cpvs could play a role in Huntington's disease because of this evidence. Firstly, in a flying model of spinocerebellar ataxia 3, it was found that ORP2, that is the ortholog of Cpv2, 3, and 4 in Drosophila, was a modulator of CAG and polyQ toxicity, and that the upregulation of this protein, ORP2, rescued abnormal wing posture and locomotor defects in this model. Also, in another study, it was found that HD-related genes are prevalent among genes that are mistranslated in the absence of Cpv1. And finally, in this work, they reported the Cpv4 binders in human pancreatic cancer cells And when we analyzed these genes, we found enrichment in HD-related genes. Together, this evidence led us to explore the status of Cpvs in Huntington's disease. In order to do that, we performed Western blot analysis on postmortem striatal tissue from individuals with Huntington's disease, and this revealed an increase in Cpv1 and a decrease in Cpv4 protein levels. We then confirmed that there was a similar alteration of Cpvs in a mouse model of HD, more precisely R61 mice, that was confirmed by immunohistochemistry. It has also been seen that Cpvs has prion-like properties, and they have polyQ-rich domains. Because of that, we check by monofluorescence if Cpv4 decrease was due to sequestration into nuclear enclosure bodies, but that was not the case. We also observed that Cpv4 does not co-localize with the Huntington positive intranuclear inclusions, so we conclude that there is an increased Cpv1 and decreased Cpv4 in brains of Huntington's disease patients and RC1 mice. Okay. Taking into account these results, we then test whether the Cpvs alteration observed in a striatum of Huntington's disease patients and mice correlates with changes in transcriptome polyadenylation. Well, for this, we extracted RNA from control and RC1 mice striatum and performed polyochromatography. This technique allows us to separate the transcripts in two fractions. One fraction have transcripts with short poly A tail, and the other fractions have transcripts with the long poly A tail. This chromatography was followed by gem chip analysis, which revealed that transcript poly A tail length increased in approximately 9% and decreased more or less in the same percentage of the analyzed genes in RC1 mice striatum. Well, we also performed gene ontology analysis and found that the three most significant terms were Huntington's, Alzheimer's, and Parkinson's disease, thus suggesting that altered polyadenylation may contribute to pathogenesis not only in Huntington's disease, but also across the main neurodegenerative disorders. In good agreement with this, genes playing key roles in neurodegeneration, such as those mutated in familial forms of Alzheimer's, tauopathies, Parkinson's, ALS, and Huntington itself, showed altered polyadenylation. This suggests a new molecular mechanism in the etiology of Huntington's disease and possibly also in other major neurodegenerative diseases. Next, we analyzed the presence of CP sequences, the one that the Cpvs bind, in the 3' untranslated region of the genes showing altered polyadenylation. What we observed was an enrichment selectively in genes showing deadenylation. This deadenylation, this shortening of the poly A tail length, is normally associated to diminished translation and decreased protein levels. To check this, we performed Western blot analysis and we confirmed reduced protein levels of the top deadenylated genes such as AUX2, ROG1, Kinectin1, both in Huntington's disease and RC1 striatal tissue, despite unaltered transcript levels. But also, among these top deadenylated genes, extremely interesting was SLC19A3 because its mutation causes biotin thiamine responsive basal ganglia disease, unveiling a new mechanistic clue about Huntington's disease. I would like to tell you more about this disease, also called BTBGD. As I said before, it's caused by mutations in SLC19A3, which encodes the transmembrane thiamine transporter 2, also called THTR2. And maybe you have heard about thiamine as vitamin B1. BTBGD is a devastating neural disorder with prominent striatal affectation that can, however, be reverted with a vitamin therapy. Individuals with BTBGD have decreased cerebrospinal fluid thiamine levels, bilateral atrophy in the head of the caudate nucleus and of the putamen, as well as a variety of neurological symptoms that some of them are very similar to Huntington's. Also, I have to say that all these symptoms improve upon administration of thiamine because it increases its concentration, and biotin because it increases SLC19A3 mRNA. We therefore hypothesize that HD might be in part a phenocopy of BTBGD due to a decrease in the transporter protein levels, and this might provide a new mechanistic clue about Huntington's as well as a potential novel therapy. Indeed, we confirm a marked decrease in the levels of this transporter in both striatum and cortex of individuals with HD without changes in mRNA. This was mirrored by a decreased staining by immunohistochemistry in both striatum and cortex, specifically in neurons and brain vessels. Before I continue, I would like to explain that thiamine has 3 forms. Free thiamine, which we acquire through our diet. This free thiamine is phosphorylated and converted to thiamine monophosphate, TMP. And the third is thiamine pyrophosphate, TPP, which comes from the phosphorylation of TMP. And is the active form of the thiamine at intracellular level because it serves as a cofactor of many enzymes, the majority of them in the mitochondria. Analyzing thiamine levels in HD patients, we found that in cerebrospinal fluid, there was a decrease in the levels of TMP, that is the prevailing form of thiamine in the CSF, despite unaltered blood levels of total thiamines, thus exactly resembling what has been reported for individuals with BTBGD. Although it has not been done in BTBGD, what is new is that we have analyzed thiamine forms in HD striatum, and we have found a marked decrease in TPP concentrations, the prevailing form of thiamine in tissue, as I have said before. These results demonstrate a thiamine deficiency in HD brain and suggest that a vitamin therapy could alleviate symptoms in HD patients. So we decided to perform a preclinical analysis. In order to preclinically test the potential of vitamin supplementation for Huntington's disease, we first confirmed that 2 mouse models of Huntington's disease, RCS1 mice and ZQ175, have the same characteristics about thiamine deficiency that Huntington's disease patients have. So, we have seen that they show decreased protein levels of the transporter, and they also show some similarities with BTBGD, such as decreased levels of glucose transport 1 in brain vessels, and also alteration in endothelial markers in brains. Also, we measured the level of thiamine forms in the striatum of these 2 HD mouse models, and we found decreased striatal concentration of TPP, as individuals with HD do. What is more important is that we found that a combined therapy of biotin plus thiamine in the drinking water of the mice prevented the decreased striatal content of TPP, as you can see in both models. Taking this into account, we decided to continue testing the potential of the vitamin supplementation. For that, we then test if the treatment was also able to improve the HD-like phenotypes in these models. Firstly, we analyzed ZQ175 mice, and we performed a magnetic resonance imaging on untreated mice to verify its striatal atrophy. And then some weeks later, and on untreated and treated mice with the biotin plus thiamine treatment, we found that the combined therapy with those two vitamins, it prevented the additional striatal atrophy experienced by untreated mice, and it even showed a tendency to slightly revert the decrease in striatal volume. Similarly, the increase in striatal phosphocreatine concentration signal seen by magnetic resonance spectroscopy was prevented by the treatment. For that, we then test if the treatment was also able to improve the HD-like phenotypes in RCS1 mice. First of all, we analyzed the motor phenotype, and we found that the motor coordination deficit detected with the rotarod test, as you can see here in RCS1 mice, was prevented by the biotin plus thiamine treatment. Similarly, the deficit in limb strength detected in the inverted grid test was also prevented by the treatment. Then we decided to analyze the neuropathology. For that, we measured the striatal area by DART32 immunostaining, and also the levels of apoptotic cells. We again found that the biotin plus thiamine treatment prevented both the striatal atrophy and the increased number of apoptotic neurons. So together, these results demonstrate that a biotin plus thiamine treatment is able to prevent the brain thiamine deficiency observed in HD mice and to attenuate their phenotype. So, in conclusion, I would like to say that we have discovered that there is a CPV4 and CPV1 imbalance in HD striatum, which led to altered polydenylation of neurodegeneration-linked genes, thus unveiling a new molecular mechanism. Also, top deadenylated genes include striatal atrophic genes and show decreased protein levels. We have seen that Huntington's disease is a BTBGD-like thiamine deficiency, and also, what is more important, we have performed preclinical studies, and we have found that a high dose of biotin plus thiamine treatment improves radiology, neuropathology, and the motor phenotype of HD mice, thus evidencing that HD patients might benefit from biotin and thiamine supplementation therapy, as BTBGD patients do. I have to say that we have published this study some weeks ago, in case you want to know more about it. And, finally, I would like to thank to all the people who contribute to this study, especially in the lab, to Jose, Parras, and Maria, who started this project a long time ago, and also the help of Miriam. I would like to thank all our contributors all around the world and our funding agencies. In case you want to ask something, just tell me a message by the chat or email, tweet, whatever you want. And many thank you.
Thank you, Sarah. That was wonderful. Thank you for the lovely presentation and all of your just fantastic work. So
Thank you.
let's see. Let me see if we have any questions. Let's see. Here's the first question. Have you checked any metabolic changes related to thymine in the mouse models?
Yes. Can you hear me?
Yes.
Okay. Yes, indeed, we have analyzed it by HPLC, and we have found an increase in some metabolites related to thiamine-dependent enzymes, such as pyruvate dehydrogenase or branch-chain alpha-cetoacid dehydrogenase complex. More precisely, what we have found are changes in the levels of some amino acids and organic acids in ARCSIS1 mice striatum. So yes, there are some metabolic changes related to thiamine deficiency.
Very interesting. So I have another question here. Have you observed if there is a direct impact of CpvB4 on the thymine transporter expression?
Yes, we have analyzed available data, and we saw that CpvB4 overexpression results in increased poly-A tail length of the SLC 1983 transcript, and we have also confirmed that crossing ARCSIS1 mice with mice overexpressing CpvB4, and we have found that there is an attenuation of the decrease in the thiamine transporter levels seen in ARCSIS1 mice, which give us a mechanistic connection between CpvB1, CpvB4 imbalance, and the SLC 1983 alteration.
Excellent, thank you. So Dr. Deborah Hall read my mind. So her question is, has someone started human-based studies yet?
Yes, we are planning to launch open label little clinical trial in order to test the security and tolerability of the treatment, and we are launching it.
Okay, lovely, thank you. And Kenneth Serban asked, in what form would HD patients take the biotin thiamine treatment? You know, pill, IV. What would you anticipate?
Yeah, yes, as BTVGD patients do, probably by taking pills, there would be the perfect treatment.
Okay. Lovely. And then let's see, we have another question here from Subrata Pradam. Is thymine treatment increased in the GLUT1 level in our 6-1 mice?
Well, we haven't tested it yet, but there is it's a good question to check, but we haven't done it yet.
And then Dr. Claudia Testa asked why open label?
Yes, first of all, the open label is because it is going to be a pilot clinical trial funded by us, and it could be a little clinical trial in order to test the security and tolerability of the treatment. If it is because the treatment will be with high doses of biotin and thiamine. If there is any problem, then we will try to enlarge the number of patients and add the placebo group. But that would be in the future.
Okay, down the road, yes, of course. There's concern that an open label study would lead to that the placebo effect may confound the results, but it makes sense. It sounds like you've definitely thought about the pros. Other questions? I think we've made it through our list of questions. I don't see anything else, any other questions that have come through. So thank you for everyone that submitted questions. And thank you again, Sara, for just a very eloquent presentation. And we look forward to hearing about your results in humans. So thank you. Thank you again.
Thank you.
Take care. Bye, everyone.